Hello everyone. Today we will demonstrate sample processing for CD34 stem cell enumeration. Before we begin, I will introduce you to our participants in workshop. She is Aswati, she is a flow trainee and she is senior fellow Dr. Anumya. For stem cell enumeration, in our lab we receive peripheral blood and apheresis product. Samples are received in EDTA or heparin vacutaneous. Once the sample are received, they should reach the lab as soon as possible at 18 to 25 degrees Celsius. Once the sample are received in lab, check for patient details and match with requisition. Check for hemolysis or clots. Clotted peripheral blood should be rejected.
We will now take the cell count. Cell count is performed to determine the WBC count. This is a cell counter. Mix the sample before taking the count. Enter the patient ID. As per the manufacturer's kit, if the count is more than 40,000 per microliter, the sample needs to be diluted. For dilution, we take 1 part of sample and 9 parts of PBS.
But why is it important to dilute the sample if the count is more than 40,000?
As per the kit's limitation, if the count is more than 40,000, it will affect the staining and bead concentration, and the results may not be reliable.
We are now done with cell count. Before we proceed to sample processing, we will discuss some basic concepts to be familiar with. We use stain, lyse, no wash method for CD34 stem cell enumeration.
But why are we not performing a wash after lysing?
This is a quantitative assay. We calculate absolute counts. We require no fixed concentration of beads and sample volume. So if we include washing steps, it will affect, you will lose your beads and sample. So this will directly affect your cell count, absolute cell count. There are two platforms for CD34 stem cell enumeration. One is single platform recommended by ICH guidelines. Second is dual platform.
What is the difference between single platform and dual platform?
In single platform, we use only one instrument, that is your flow cytometer. In dual platform, there are two instruments involved. One is your flow cytometer and the other is WBC counter. So you get your CD34 percentage from the flow cytometer and WBC count from cell counter. So the WBC count from the cell counter, it is used as denominator, but the count may not be reliable. It will affect your absolute count. In single platform, we use beads to determine the absolute counts. We will now discuss the assay principle for CD34 stem cell enumeration by single platform method. Sample is first added to the fluorochrome label antibodies. The fluorochrome label antibodies in the reagent bind specifically to the cell surface antigens. 7-AD dye is added to assess the viability of cells. Cells that are 7-AD positive are not viable. Ammonium chloride based lysing solution is added to lyse RBC. Known concentration of beads is added to get absolute counts of CD34 stem cells.
Commonly used kits for CD34 stem cell enumeration are from Beckman Coulter and BD Biosciences. These are stem kit reagents provided by Beckman Coulter. The kit includes 1. Monoclonal antibody cocktail of CD45 FITC conjugated and CD34 P conjugated, specific for different cell surface antigens. 2. 7-AD viability dye, that is 7-aminoactinomycin dye to distinguish between viable and non-viable cells. 3. CD45 FITC conjugated isoclonic control P to check non-specific binding of CD34P monoclonal antibody. 4. 10x ammonium chloride based lysing solution for RBC lysing. For working, use freshly prepared 1x solution diluted using distilled water. Last one, stem count fluorospheres in liquid form. This reagent contains beads having known concentration used for absolute counting. The beads are excited at 488 nanometer with emission range between 525 to 700 nanometer. Important to note that stem count fluorospheres available in liquid form should be mixed properly before use to avoid alteration of bead concentration. These are stem cell enumeration kit reagents provided by BD Biosciences. The kit includes 10x ammonium chloride based lysing solution for RBC lysing. For working, use freshly prepared 1x solution diluted using distilled water. Second is 7AD viability dye to distinguish between viable and non-viable cells. Third, BD stem cell reagent that contains monoclonal antibody cocktail of CD45 FITC conjugated and CD34 P conjugated, specific for different cell surface antigen. BD provides true count tube. The tube contains freezed, dried, lyophilized pellet of fluorescent beads that dissolve, releasing known number of fluorescent beads. True count tubes has an advantage compared to using Beckman-Coulter stem count fluorospheres in liquid form. Pipetting errors may affect absolute stem cell counts.
We will now process the sample. The sample is processed in duplicates.
But why are we processing in duplicates?
The sample is processed in duplicates to identify technical errors during processing, such as pipetting, RBC lysing, and inappropriate sample mixing. We will now label the tube with the patient ID. Add 20 microliter of CD4534 cocktail to both the tubes. Add 10 microliter of 7AD dye to both tubes. Now we will add 100 microliter of sample by reverse pipetting.
What is reverse pipetting and why do we do reverse pipetting?
Reverse pipetting, it is a technique to dispense measured volume and in precise manner, which reduces splashing, foam and bubble formation that will affect your absolute counts. I will now demonstrate reverse pipetting to you. Hold the pipette. This is the plunger. The pipette has two stop levels. First stop, second stop. Okay. The sample is aspirated at the second stop. The sample is dispensed at the first stop and then release the plunger. Do you want to try?
Yes. So I will aspirate the sample while pressing the plunger till the second stop and aspirate, and while dispensing I will press the plunger only till the first stop, will release the plunger and then discard the tip.
Mix the sample properly. Aspirate 100 microliter by reverse pipetting. Press the plunger to second stop. Aspirate the sample. Wipe the outside wall of the tip. Avoid touching the end of the tip to the tissue paper.
To avoid capillary action of tissue.
Dispense the sample at the first stop. Release. Avoid touching to the wall. Discard the tip. We will again add sample to the second tube. Mix the sample before addition. Press the plunger to the second stop. Aspirate the sample. Wipe the excess blood from the walls of the tip. Avoid touching the end of the tip. Dispense the sample at the first stop. Release. Avoid touching to the walls. Now we have added the antibody cocktail and the sample vortex. Incubate for 20 minutes at room temperature in dark.
Now I will show you difference in normal pipetting and reverse pipetting. In normal pipetting, sample is aspirated at first stop. When you aspirate sample at first stop, you aspirate exact volume of sample. Sample is dispensed at second stop. Some volume of sample is left in the tip. You lost this sample. This will affect your absolute cell count. When you try to dispense this remaining volume, this will create bubble formation. In reverse pipetting, sample is aspirated at second stop. When you aspirate sample at second stop, you aspirate excess volume of sample. Sample is dispensed at first stop. You dispense exact volume of sample.
Meanwhile, we will prepare fresh 1x lysine solution. This is 10x lysine solution. We have to take 1 part of lysine solution and 9 parts of distilled water. For 1 tube, we require 2 ml. We will prepare total 5 ml of lysine solution. I will take 500 microliter of 10x lysine solution. And dilute it with 4.5 ml of distilled water to get 1x concentration. Incubation is over. Vortex the tubes. Add 2 ml of 1x lysine solution to both the tubes. Vortex the tubes. Incubate for 10 minutes in dark at room temperature. Now the tubes are ready for acquisition. Vortex before acquisition.